Hello students, this is Dr. Tikia Arashiro, and in this podcast, I'm going to talk about lithotrophy, which is a type of inorganic metabolism. While most organisms derive energy from oxidation of organic compounds or from absorption of light, there exist many prokaryotes that derive energy from the oxidation of inorganic compounds or molecules. These organisms are called lithotrophs. Examples of inorganic compounds or molecules include hydrogen gas, carbon monoxide, carbon dioxide, ammonia, nitrite, hydrogen sulfide, elemental sulfur, thiosulfate, and ferrous iron. Lithotrophy or chemoautotrophy is the acquisition of energy by oxidation of inorganic electron donors. It is an oxidative inorganic metabolism. The inorganic compounds are used as electron donors, which are oxidized to generate energy. Lithotrophy is restricted to prokaryotes and does not occur among eukaryotes. It is a catabolic process. The organisms are called lithotrophs or chemoautotrophs. Lithotrophs are physiologically diverse and exist among several groups of bacteria and archaea. They have a diverse respiratory growth. Most of the lithotrophs are aerobes and carry out electron transport with oxygen as electron acceptor. However, some are facultative anaerobes using nitrate or nitrite as electron acceptor when oxygen is unavailable, and a few are obligate anaerobes that use sulfate or carbon dioxide as electron acceptor. For most lithotrophs, the sole or major source of carbon is carbon dioxide. Such organisms are called chemolithoautotrophs. Now, the lithotrophs vary in terms of the autotrophic carbon dioxide fixation pathway they use. Other lithotrophs are facultative heterotrophic and are called facultative heterotrophs. Now, facultative heterotrophs can either use carbon dioxide or organic carbon as the carbon source. So they are sometimes called mixotrophs, meaning they have the ability to use both heterotrophic methods as well as autotrophic methods. So prokaryotic microorganisms that carry out lithotrophy are called lithotrophs or chemoautotrophs. Some representative species are listed on this table. Some lithotrophs are aerobes and use oxygen as electron acceptor, such as ammonia oxidizing prokaryotes and nitrite oxidizing prokaryotes. Some are strictly anaerobe and use carbon dioxide as an electron acceptor, like the methanogenic prokaryotes and the acetogenic prokaryotes. Some are facultative anaerobes that use nitrate when oxygen is unavailable, like hydrogen oxidizing prokaryotes, the carbon monoxide oxidizing prokaryotes, the sulfur oxidizing prokaryotes, and the iron oxidizing prokaryotes. For most lithotrophs, the carbon source is carbon dioxide. However, other lithotrophs are facultative heterotrophs, meaning they can grow autotrophically and heterotrophically. So I'm going to talk about each of this in the following slides. The first group of lithotrophs that I'm going to talk about are the hydrogen oxidizing lithotrophs. Hydrogen oxidizing lithotrophs are lithotrophs that oxidize hydrogen gas to water. Most are aerobic and use oxygen as electron acceptor. Hydrogen oxidizing lithotrophs are facultative heterotrophs and can live either autotrophically using carbon dioxide as carbon source and heterotrophically using organic carbon as carbon source. However, some always require organic carbon for growth, and these are called chemolithoheterotrophs. Chemolithoheterotrophs are a special kind of chemotroph that use inorganic compounds as energy source and reduce organic compounds as carbon source. Now, hydrogen oxidizing lithotrophs are mostly aerobic, although some are anaerobic, such as the methanogens and sulfate-reducing bacteria that are capable of using hydrogen gas as electron donor. And because they are anaerobic, they do not use oxygen as electron acceptor. Methanogens use carbon dioxide as electron acceptor, while sulfate-reducing bacteria use sulfate as electron acceptor. So where can we find hydrogen oxidizing lithotrophs? Hydrogen oxidizing lithotrophs can be found in aerobic and anaerobic environments where hydrogen gas is available. Recall that hydrogen gas itself is produced as a byproduct of nitrogen fixation in plants and rhizobium, in cyanobacteria, and other nitrogen fixers. So you'll often find hydrogen oxidizing bacteria in environments where nitrogen fixers are hanging out. Hydrogen gas is also produced in anaerobic environments, such as during fermentation, and they escape into the aerobic atmosphere and eventually taken or used as electron donor by hydrogen oxidizing organisms. Now, hydrogen oxidizing lithotrophs oxidize hydrogen gas to water, so the end product of hydrogen oxidation is water. The second group of lithotrophs are the carbon monoxide oxidizing prokaryotes or carboxylobacteria. These organisms oxidize carbon monoxide to carbon dioxide. They utilize carbon monoxide both as carbon source and energy source. Note that carbon monoxide is found in the atmosphere. Carbon monoxide oxidizing bacteria are facultative anaerobes and use oxygen as electron acceptor when it is available and nitrate as electron acceptor when oxygen is unavailable. They are chemolithoheterotrophs or facultative heterotrophs and can either use inorganic carbon like carbon dioxide or organic carbon like glucose and methanol as carbon source. Carboxylobacteria oxidizes carbon monoxide to carbon dioxide, so the end product of carbon monoxide oxidation is carbon dioxide. The third group of lithotrophs are the ammonium oxidizing prokaryotes. Nitrosomonas europaea is an ammonium oxidizing bacterium, and Nitrosopamilus maritimus is an ammonium oxidizing archaeon. They use ammonium as electron donor. They are strictly aerobic and use oxygen as electron acceptor and oxidize ammonium to nitrite. So the end product of ammonium oxidation is nitrite. The oxidation of ammonium to nitrite generates energy in a form of ATP. Ammonia oxidation involves a 6-electron transfer. This means that 6 electrons must be lost from ammonium to oxidize it to nitrite. The oxidation state of ammonium is -3 and the oxidation state of nitrite is +3. So +3 minus -3 is equal to 6. So 6 electrons will have to be taken away from ammonium to oxidize it to nitrite. This image here shows you the electron transport system of ammonia oxidizing bacteria, Nitrosomonas. The arrangement of the electron carriers in the electron transport system of Nitrosomonas are speculative and are based on known enzymes and their redox potential. Although the oxidation of ammonia is depicted as occurring in the cytoplasm, the possibility that it occurs in the periplasm has not been ruled out. Hence, ammonia oxidation can occur either in the cytoplasm or the periplasm. The key enzymes in the oxidation of ammonia to nitrite are the ammonia monooxygenase and the hydroxylamine oxidoreductase. Ammonia monooxygenase is a metalloenzyme that catalyzes the oxidation of ammonia or ammonium to hydroxylamine. So ammonium here reacts with the electron acceptor oxygen and becomes oxidized to hydroxylamine. The first oxidation step requires the removal of 2 electrons from ammonium. So 2 electrons have to be removed in the conversion of ammonium to hydroxylamine. Hydroxylamine oxidoreductase then catalyzes the conversion of hydroxylamine to nitrite. 4 electrons have to be removed in the conversion of hydroxylamine to nitrite. So ammonia monooxygenase, shown here as AMO, is a membrane protein, while hydroxylamine oxidoreductase, shown here as HAO, is a peripheral protein. They are components of the electron transport system in ammonia oxidizing prokaryotes. So here's the plasma membrane of nitrosomonas showing the electron transport system. Ammonium enters the cell via a transfer protein. Once in the cell, ammonium monooxygenase oxidizes ammonium to hydroxylamine. This reaction is oxygen dependent and 2 electrons are released from ammonium in this process. Hydroxylamine then crosses the plasma membrane where it is oxidized to nitrite by the enzyme hydroxylamine. 4 electrons are released in this reaction. The fourth group of lithotropes are the nitrite oxidizing prokaryotes. Nitrite oxidizing prokaryotes are aerobic chemoautotropes. They use nitrite as electron acceptor and carbon dioxide as carbon source. Nitrite oxidizing prokaryotes oxidizes nitrite to nitrate. So the end product of nitrite oxidation is nitrate. Now, nitrite oxidizing prokaryotes work hand in hand with ammonia oxidizing bacteria to complete the nitrification process. Nitrification is the biological oxidation of ammonia to nitrate and is primarily accomplished by 2 groups of lithotropes, the ammonia oxidizing bacteria and the nitrite oxidizing bacteria. Together, they convert ammonia to nitrate. For example, the ammonia oxidizing bacterium Nitrosomonas first oxidizes ammonia to nitrite, then the nitrite oxidizing bacterium Nitrobacter oxidizes nitrite to nitrate. The first oxidation is that of ammonia to hydroxylamine, catalyzed by ammonia monooxygenase, or AMO. In this reaction, 1 oxygen atom is incorporated into hydroxylamine and the other is reduced to water. The oxidation of hydroxylamine to nitrite is catalyzed by the enzyme hydroxylamine oxidoreductase, or HAO. The nitrite is oxidized to nitrate by the nitrite oxidizing bacterium Nitrobacter. The key enzyme in the oxidation of nitrite to nitrate is the enzyme nitrite oxidase. This reaction is oxygen dependent. The nitrite oxidizers include Nitrobacter, Nitrococcus, Nitrospina, and Nitrospira. They are aerobic obligate chemolithotropes with the exception of Nitrobacter, which is a facultative heterotroph. The details of the electron transport scheme have not been fully elucidated. However, a proposed model is shown in this figure. The key enzyme in nitrite oxidation is the nitrite oxidase, which is a membrane-bound enzyme. The electrons travel from nitrite via nitrite oxidase to cytochrome C. It is proposed that nitrite oxidation takes place in the cytoplasm or the cytoplasmic side of the membrane, and the electrons flow across the membrane to cytochrome C located on the periplasmic side of the membrane. For clarification, this is the cytoplasmic side of the membrane and this is the periplasmic side. 2 electrons have to be removed from nitrite to oxidize it to nitrate. Now, from cytochrome C, the electrons diverge. Most travel to oxygen at a more positive potential, so the electrons flow from cytochrome C back across the membrane to oxygen through cytochrome AA3 oxidase, which is a proton pump. Cytochrome AA3 oxidase reduces oxygen to water. Now, the electron flow can also be reversed from cytochrome C to NAD+, so other electrons travel to NAD+, which is at a more negative potential. They pass through 2 coupling sites, a ubiquinone cytochrome C oxidoreductase and a reverse NADH ubiquinone oxidoreductase. The NADH ubiquinone oxidoreductase reduces NAD+ to NADH. The 5th group of lithotropes are the sulfur oxidizing prokaryotes, or sulfur oxidizers. Sulfur oxidizers oxidize various forms of sulfur to sulfate. These include hydrogen sulfide, elemental sulfur, and thiosulfate, and these are used as electron donors by sulfur oxidizing prokaryotes. Now, there are 2 groups of sulfur oxidizers, the chemoautotrophic sulfur oxidizers and the photoautotrophic sulfur oxidizers. Both of these utilize inorganic sulfur, such as hydrogen sulfide, elemental sulfur, and thiosulfate as electron donor. In sulfur oxidation, The electron donor sulfur is oxidized to sulfate. Sulfur oxidizers comprise a physiologically heterogeneous group. They may be obligate autotrophs, which grow only on carbon dioxide as the carbon source, or facultative heterotrophs, which can also use organic carbon as the source of carbon. Some are neutrophiles, growing best around pH 7, whereas others are acidophiles, growing best between pH 1 and 5. Sulfur oxidizing acidophiles can be isolated from sulfur mines which produce sulfuric acid. An example is Acidithiobacillus thiooxidans, which can grow at a pH of 1, although the optimum is between 2 and 3. Some oxidizing acidophiles can be mesophilic, with growth temperature optimum between 25 and 40 degrees Celsius, or thermophilic, with growth temperature optimum greater than 55 degrees Celsius. Sulfur compounds commonly used as sources of energy and electrons include hydrogen sulfide, elemental sulfur, and thiosulfate, all of which can be oxidized to sulfate. Now, the oxidation of the thiosulfate to sulfate is a 4-oxidation step. 4 electrons will have to be removed from thiosulfate to oxidize it to sulfate. There has been much confusion in the literature regarding the intermediate stages of sulfur oxidation. Probably more than one pathway exists. 2 pathways that are receiving much research attention are those mediated by the sulfur oxidizing bacterium Paracoccus virsutus and the sulfur oxidizing bacterium Thiobacillus tepidarius. In this podcast, I'll be focusing on the pathway mediated by Paracoccus virsutus, which is shown in this figure. Paracoccus virsutus is a neutrophilic facultative lithoautotroph. It can grow not only on thiosulfate, but also on various organic carbon sources. This organism was formerly called Thiobacillus virsutus. Oxidation of thiosulfate, which is the electron donor, takes place in the periplasm on a multi-enzyme complex. So here is the cytoplasm and here is the periplasm. So 2 molecules of thiosulfate are oxidized to 2 molecules of sulfate in the periplasm. No free intermediates are released and both molecules of thiosulfate are oxidized to 2 molecules of sulfate. Protons and electrons are released in the periplasm during the oxidation, and the 8 electrons are transferred electrogenically from the periplasmic side of the plasma membrane to the cytoplasmic side through cytochrome C552 and then cytochrome AA3 oxidase to oxygen. The cytochrome AA3 oxidase reduces oxygen to water. A proton current is maintained by the release of protons in the periplasm using the oxidations and their consumption in the cytoplasm during the reduction of oxygen. The 6th group of lithotrophs are the iron-oxidizing prokaryotes. Iron-oxidizing prokaryotes derive their energy from the oxidation of ferrous iron to ferric iron. They are assumed to be obligately aerobic using oxygen as electron acceptor, but iron-oxidizing prokaryotes are facultative anaerobes. When oxygen is not available, they use elemental sulfur as electron donor and ferric iron as electron acceptor. They oxidize the electron donor elemental sulfur to sulfate and reduce the electron acceptor ferric iron to ferrous iron. Most of iron-oxidizing prokaryotes are also acidophilic sulfur oxidizers, so they can also oxidize sulfide to sulfuric acid. An example is Thiobacillus ferrooxidans, which is an autotroph able to use either ferrous iron or inorganic sulfur compounds as source of energy and electrons. Thiobacillus ferrooxidans can be grown on ferrous iron like ferrous sulfate at an optimal external pH values between 1.8 and 2.4. Since ferrous iron is rapidly oxidized chemically by oxygen to ferric iron at neutral pH, but only slowly at acid pH, the acidic environment is conducive to the growth of the iron oxidizers. This figure illustrates a model for the electron transfer pathway in Thiobacillus ferrooxidans growing on the aerobic oxidation of ferrous sulfate. Here is the outer membrane of Thiobacillus ferrooxidans, here is the periplasm, here is the plasma membrane, and here is the cytoplasm. In this proposed pathway, the extracellular ferrous iron is oxidized to ferric iron by ferric iron complex in the outer membrane. The electrons travel to a periplasmic cytochrome C. The periplasm also contains a copper protein called rustacyanine, which is thought to be a part of the electron transport scheme. RC here stands for rustacyanine and C stands for cytochrome C. From cytochrome C, the electrons flow across the plasma membrane via cytochrome oxidase or cytochrome A1 to oxygen. The electrons are then transferred to the electron acceptor oxygen, which is subsequently reduced to water. So the oxidation of ferrous iron drives the consumption of protons in the cytoplasm during oxygen reduction to water. The consumption of 2 protons for every 2 electrons in the cytoplasm and the uptake of 3 protons through the ATP synthase indicate that a maximum of 2/3 of an ATP can be made for every oxidation of 2 ferrous irons. So that concludes this podcast on inorganic metabolism. Thank you for your attention. I hope you enjoyed watching and listening to this podcast. If you want to read more about this topic, you can refer to the sections from Chapter 13 of the recommended text, The Physiology and Biochemistry of Prokaryotes.